Hi everyone, this is lecture 21 in the second part of the digestive system. Here we're going to continue on past the esophagus and start with the stomach and its structure and functions and some of the accessory organs in the small intestine. So we're going to continue with the processes that are involved in swallowing. We'll talk about the structure and function of the stomach and some of the functions of the pancreas, liver, and gall bladder. Okay. So first things first, deglutition is another the clinical term for swallowing. And so it's the propulsion of food from the pharynx to the esophagus to the stomach. And next time you're eating, think about what's happening as you swallow. It's a coordinated action of different parts of your oral cavity and esophagus, including the soft palate, pharynx, and the tongue. Deglutition, or swallowing, is composed of three phases: a voluntary phase, a pharyngeal phase, and an esophageal phase. In this figure shows the three processes. The voluntary phase is named because it's under conscious control of the cerebral cortex, and so you have the ability to swallow or not to swallow at this point. And the tongue pushes the bolus posterior toward the oral pharynx. In the second step, the pharyngeal phase, the bolus enters the oral pharynx, and the soft palate and the epiglottis seal off the nasopharynx and larynx, respectively. And the third esophageal phase, peristaltic waves moves the bolus down the esophagus to the stomach. So here are the three phases of swallowing or deglutition. We have the voluntary, pharyngeal, and the esophageal. This shows you the control for each. We said the voluntary phase is under the cerebral cortex, and you have conscious control. The pharyngeal phase is under control of the medulla and the enteric nervous system. The esophageal phase is under control of the medulla and the autonomic nervous system. The stomach is a J-shaped organ after the esophagus, and it has sphincters at either end. So here we see the stomach here, the J-shaped organ, and we see that there is a gastroesophageal sphincter separating the esophagus from the stomach, and a pyloric sphincter that's separating the end of the stomach to the first part of the small intestine, or the duodenum. Inside the stomach, it's not smooth, but it contains many folds called ruga, allowing it to expand in volume. So you can see the rugae here on the lining of the stomach. Now, the stomach has many functions. We can divide them into three basic parts: secretion, propulsion, and digestion. And we will see that the stomach's structure is closely related to its function. So what's different about the histology of the stomach? Well, one of it has a modified Muscularis, Externa, and Mucosa, because it has an extra inner oblique layer of smooth muscle. So it has a third layer of smooth muscle. In addition to the circular and longitudinal, it has an oblique layer. And this is present because it allows the stomach to churn the food, to help breaking it up mechanically into liquid chyme. So here is showing the outer longitudinal layer, the middle circular layer, and the inner oblique layer. Now the stomach itself has what's called gastric pits, and these are indentations of the stomach mucosa. Okay, and in reality though, this figure is showing you the pits, but they're showing you the sort of the extensions between the pits. Okay, and between the gastric pits secrete mucus to protect the stomach. So the stomach is protected from digestion by this mucus layer. Okay, and so here are many goblet cells mixed amongst the simple columnar epithelial cells that secrete the mucus that fills the gastric pits. Okay. Now, there's also gastric glands present in the stomach, and this secretes an acidic enzyme fluid called gastric juice, and it's composed of 4 different cell types. Okay, we'll take 2 at a time. First are mucous neck cells. These secrete an acidic mucus. We have the parietal cells, which are very important because they secrete the hydrochloric acid, and the hydrochloric acid is important because it activates an enzyme called pepsinogen, which is a protease, and the HCl also destroys bacteria. The parietal cells also secretes what's called intrinsic factor, which will be important for vitamin B12 absorption. On this figure here we see the mucous neck cells, near the bottom of the gastric pits, and here we're showing the parietal cells. The second 2 cell types found amongst the gastric glands are the chief cells, which secretes pepsinogen, which is the inactive enzyme that HCl will act on. Also, a group of cells called diffuse neuroendocrine cells. Okay, there's just a mixture of different types of cells that secretes hormones for digestion. For example, we have G cells that secretes gastrin, which stimulates secretions from the parietal cells. So here in this figure, we see the chief cells, and we see the enteroendocrine cells here in blue here. So here are endocrine cells, for example, and the chief cells. Chief cells, sorry. So here the chief cells are secreting pepsinogen, which is activated by the acidic pH produced by the HCl, and which becomes the enzyme pepsin. Okay, so we'll talk more about these proteases in a little bit later lecture. Remember, but for now we'll say pepsinogen is an inactive enzyme, which is cleaved by HCl and activated to the active enzyme pepsin. Here we also show the neuroendocrine cells called G cells that secrete the hormone gastrin, which stimulates the secretions from the parietal cells. So the question is, why don't parietal cells become digested by HCl they produce? If it's strong enough to digest bacteria, why doesn't it digest the intestinal lining? Well, the reason why is because the hydrochloric acid is formed outside the cell, so the cells are actually protected. And we can see how this happens because hydrogen ions are secreted by an ATP-dependent hydrogen-potassium pump, which secretes hydrogen ions moving out of the cell, whereas potassium is moved into the cell. So this is an exchanger which exchanges hydrogen ions with potassium. Now, chloride follows the hydrogen ions via passive diffusion through a channel. Now you have hydrogen ions and chloride ions outside the cell to produce the HCl. Okay. So we see in this picture here a parietal cell, which shows the hydrogen ion potassium pump, which is active transport, noticed by the ATP here. So for every hydrogen that moves out, one potassium moves in. And through the charges, chloride falls passively through its own channel. And so now you have the HCl, hydrogen ions and chloride ions produce HCl outside the cell, protecting the parietal cells from the HCl. Because here along here is the mucous lining from the gastric pits. The second major function of the stomach is propulsion. The stomach has motility, and the motility is used for three different purposes. One, it has to receive food from the esophagus, and it has to churn the bolus into chyme. Okay, this bolus of food has to be kind of liquefied into chyme. And finally, it has to control the rate that the chyme empties into the small intestine. It can't deliver it all at once, but it has to give it, it sets the rate of how much of the chyme empties into the duodenum. So let's take the first function first, the receptive function. We saw early on that there are sphincters that allow, called the gastroesophageal sphincter, that relaxes and allows the stomach to fill. So when the stomach's empty, it holds about 50 mils, but can expand to hold up as much as 1,500 milliliters. And the stretching of the rugae relaxes the smooth muscles of the stomach wall to help it expand. The churning function is due to the peristaltic waves of smooth muscle layers of the stomach. Remember, we said that there was a third layer of the stomach wall that allows it to churn in 3 dimensions. We have the circular layer, the longitudinal layer, and now we have the oblique layer. And all 3 help for the churning function. And so in this diagram, kind of shows you the peristaltic action of these muscles. So here it's showing the smooth muscle contracts and propels a small amount of chyme through the pyloric sphincter. Okay, the rest is propelled backwards. So it's only allow a small amount of the chyme into the duodenum. Then the peristaltic action of the stomach continues to churn the remaining chyme, and the chyme is propelled forward to the pyloric sphincter again, and then more is forced through as the process repeats. So it only periodically allows so much of the chyme into the duodenum, a little bit at a time, so that there's adequate churning to form the chyme in the stomach. Okay, so finally, the emptying function of the stomach. It controls the movement of chyme into the duodenum, and materials pass at different rates depending on the amount and composition of the chyme. So different factors such as high stretch, low pH, high lipid content of the food, high soil concentration will delay gastric emptying. Okay, so these will delay gastric emptying. And of course, it has to set the rate, because the duodenum cannot take all the chyme at one time. It takes time to process the material. So, in addition, the acidic chyme from the stomach has to mix with bicarbonate from the duodenum to prevent damage, because remember, the stomach is protected by the mucous lining, but the small intestine does not have that mucous lining. We're going to see later that one of the major functions of the small intestine is for reabsorption. Of course, having a mucous lining would kind of slow absorption up, would slow absorption. So the small intestine will have to mix bicarbonate with the acidic chyme to protect it. It also dilutes chyme with pancreatic juice, so that the solids are at a lower concentration to prevent drawing water by osmosis. Okay, so let's talk about the small intestine. It's the longest part of the digestive system. It's only called small because it's a small diameter. And it has 4 important processes: secretion, digestion, absorption, and propulsion. Okay, because we saw that it has to secrete many substances, such as bicarbonate, pancreatic juice. It's going to be in the area of major digestion in the digestive tract. And of course, this is going to be a major area where the nutrients are absorbed. And of course, it has to move it throughout the small intestine and then move to the large intestine. There are 3 divisions of the small intestine: the duodenum, the jejunum, and ileum. The duodenum begins at the pylorus and is the shortest division, maybe only like 10 centimeters. It's interesting because it's approximately intraperitoneal, but posteriorly retroperitoneal. So you can have a single structure that is both intraperitoneal and retroperitoneal. Okay, remember these terms. Intraperitoneal is within the peritoneal cavity, and retroperitoneal is outside of it. Remember that the pyloric sphincter regulates the movement of the chyme from the stomach. Okay, so here's the stomach with the pyloric sphincter, and we see the small intestine kind of stretched out a little bit to make it easy to see. And we see the different regions, the duodenum, the jejunum, and the ileum. The jejunum is the middle portion, is about 2.5 meters. Okay. It's going to be the active site for chemical digestion and absorption. Okay, so this is where most of the action of digestion and absorption occurs. So here in green is the jejunum from here to here. The ileum is the last part of the small intestine and is the longest, about 3.6 meters. And it ends at the cecum, where it joins the large intestine. And as you might expect, the ileum has what's called the ileocecal valve, which controls the movement from the ileum into the cecum. So here in yellow is the ileum. Notice the spelling is different from the bone ileum. Here instead of the E. And here's the ileocecal valve that separates it from the small intestine from the cecum of the large intestine. So let's look at the structure and function of the small intestine. One unique structure of the small intestine is that it has internal folds, and these internal folds will increase the surface area for absorption. Okay. By having a larger surface area, there's more areas that can be used for bringing nutrients across the GI tract wall. So the first of these are called circular folds, or plicae circularis, and these are the largest of the mucosa and submucosa. They're like little speed bumps. They slow movement of chyme to allow time for digestion and absorption to occur. Okay, if it goes too fast, you're not gonna get any digestion or absorption. So it has to go slow enough for both of these processes can occur. Okay, here's a picture showing the lining of the small intestine, and you can see these folds. Okay, and these are circular folds, or plicae circularis. Another projection are called villi. Okay, and villi are projections of mucosal folds. Okay, each lined with enterocytes of simple squamous epithelium. Okay, so here is showing the villi of simple squamous epithelium, and you can see how they can easily increase surface area of the small intestine. Now, each has goblet cells. Okay, so you know it secretes mucus. Okay, it's surrounded by capillaries, because remember reabsorption is going to occur, and also lacteals. Do you remember lacteals? These are lymphatic vessels in the small intestine, and these are going to help absorb fats. You might have learned about these in SCB 203. And we also have what are called intestinal crypts that contain enteroendocrine cells. Okay, so here in the villi we have the goblet cells that secrete mucus. We have the lacteals that are going to absorb fats. We have blood capillaries that are going to absorb other things that are reabsorbed or absorbed. And as well, we have these intestinal crypts that are lined with these enteroendocrine cells. Third, we have what are called microvilli. Okay, from the name, these are going to be much smaller than a villi. And these are the smallest folds in the plasma membranes of the enterocytes. They look so hair-like that it appears like a brush border, and they're given this name as the brush border of the small intestine. And due to the increase in surface area, many digestive enzymes are produced and secreted by the enterocytes. Okay, so here's the villi, and if we expand an area on the end of one villus, you see that each is covered by microvilli, and from the viewpoint from the outside, it will look like a brush border. So let's talk about motility of the small intestine. Remember that propulsion is going to be, movement is going to be another function of the small intestine. And it differs whether you're fasting or eating. Okay? So if you're fasting, motility is going to be slow. You have slow rhythmic contraction, which makes sense because there's not going to be anything there to move. It takes about 2 hours to move digested food from the duodenum to the ileocecal valve. It's controlled by the enteric nervous system, and also another hormone called motilin, which is nicely named from the duodenum mucosa. Now, motility during feeding, you have two different types of movement. One's called peristalsis, which we mentioned before, and also another one called segmentation, which is regulated by the vagus nerve. Peristalsis is alternating contractions by the longitudinal and circular layers of the Muscularis externa. So now back in the small intestine, we're back down to two smooth muscle layers. Remember the stomach had that third layer, the oblique layer? Well, the small intestine is back down to the circular and longitudinal layers. So that oblique layer is only in the stomach. And this peristalsis is what propels the chyme to the cecum. Okay, so on the left here is demonstrating peristalsis, showing the alternating contractions of the circular muscle, okay, and contraction of the longitudinal muscle. So the circular muscle will kind of squeeze it here, and the longitudinal muscle will kind of propel it forward. The other type of movement during feeding is called segmentation. Another term is called intestinal churning. And this one only involves the circular layer of the smooth muscle. And this squeezing motion mechanically digests and mixes chyme with the intestinal and pancreatic enzymes, and also bile that's going to be entering the small intestine, which we'll talk about in a little bit. And so here in segmentation, the mixing contractions of the circular muscles only will help move the fluid with the chyme and mixing it all up. And so you can see the circular actions by these arrows. So let's talk a little bit about the accessory organs of the digestive system now. And it's a good place because this is where many of the secretions are entering the small intestine. Because remember, the small intestine is where there's going to be much of the digestion and absorption. So we'll talk about the pancreas, the liver, and the gall bladder. And all these, since they're emptying into the lumen of the small intestine, function as exocrine glands. Okay, because remember, exocrines secrete via a duct to outside the body. So the lumen of our digestive system is still considered outside of our body, even though it's internal. And so these are served by exocrine glands. Now, pancreas we talked about already. We covered the endocrine system. So the pancreas is kind of unique because it has both endocrine as well as exocrine functions. When we talk about the exocrine functions, we're talking mostly about enzymes that are secreted by the acinar cells that help digestion. So they're going to be important for the chemical digestion of the chyme. This is a little bit the structure of the pancreas. It's located in the upper left quadrant of the abdomen, and it's mostly retroperitoneal. Okay. And as you can see, it's composed of a head, body, and tail. Okay. It's served by the main pancreatic duct, which receives secretions from the acinar cells. The main pancreatic duct is also joined by the common bile duct from the liver and gallbladder, or separately as the accessory pancreatic duct to empty into the duodenum. So we here see the main pancreatic duct here. It's joined by the common bile duct that's coming from the gallbladder and liver up here. Or it can also enter separately through the accessory pancreatic duct. The only difference is that the accessory pancreatic duct will only have pancreatic secretions, whereas this main pancreatic duct will have secretions from both the pancreas as well as the liver and gallbladder. Now, the fluid that's produced by the pancreas is called pancreatic juice. And it's sort of the collection of all the secretions of the acinar and duct cells. The major component of the pancreatic juice, as you might expect, is water, many different types of digestive enzymes, as well as proteins. Okay? Another component of pancreatic juice, which we talked about earlier, is bicarbonate. And remember that bicarbonate acts as a buffer to make the pancreatic juice alkaline. Okay? Remember the alkalinity is important because the bicarbonate will protect the duodenum by neutralizing the acidic chyme that's coming from the stomach. Because the small intestine is not as protected as the stomach. Okay, so here's a picture of the pancreatic acinar cells secreting their pancreatic components in the pancreatic juice. Here's bicarbonate ions being secreted, as well as enzymes entering the main pancreatic duct into the duodenum. So. Let's talk about the liver and the gallbladder. The liver is inferior to the diaphragm. That's why it's kind of dome-shaped. And the gallbladder is a small sac on the liver's posterior side. There are 4 lobes to the liver: the right, left, caudate, and quadrate lobe. The latter 2 are kind of smaller because when you're looking at the anterior view, you primarily just see the right and left lobe. You have to look underneath to see the quadrate and caudate lobe. The liver histology is kind of unique, okay, and because they're composed of lobules, and the cells of the liver are called hepatocytes. So here's a section of a liver, and we can see here's a single liver lobule here, and you can see it has almost a geometric shape, and they're composed of cells called hepatocytes. Now, between the lobules is a central vein, and each corner has a portal triad that contains a hepatic arteriole, a portal venule, and a bile duct. And the capillaries are called the hepatic sinusoid because they're very leaky. Okay, so you see in this geometric shape of the lobule, we have a central vein. In each corner, we have a lymphatic vessel, an arteriole, and a venule. And then within it, we have these hepatic sinusoids, which are the leaky capillaries. Now, the liver has many functions. We're primarily concerned in its digestive function, but we'll name them all for now: bile production, nutrient metabolism, detoxification, as well as excretion. These are the functions of the liver. However, bile production is the major digestive function of the liver. And bile has important functions. One, it's needed for the digestion and absorption of lipids. Okay, notice the wording here. It's needed for digestion. It itself does not digest, absorb, but bile is required so that the enzymes can digest the lipids. The bile also proves a way to remove waste the kidneys cannot excrete. Okay, bile salts are the main component of bile, and it originates from cholesterol. And bile salts are an amphipathic molecule. So the cholesterol framework is hydrophobic, but since they're salts, they also have a hydrophilic end. So remember, when a molecule has both a hydrophobic end and a hydrophilic end, we call them an amphipathic molecule. And they kind of act like almost like a detergent, almost. So that, like for example, soap will help remove charged dirt molecules away. So what bile salts do is they coat the lipids surrounding them, and they break apart these fat lipid molecules into smaller ones by emulsification. Okay, So notice the wording here. We say break apart rather than digestion. Okay, so they do not digest fats, but they break them apart. Okay, but bile itself all contains cholesterol, waste products, and toxins that pass into the feces. Okay, the gallbladder itself is just a storage organ for the bile. Okay, it receives most of the bile via the common hepatic duct, and so it stores bile. It releases bile when stimulated by a hormone called cholecystokinin, CCK, by the duodenal cells. Okay, so here's the gallbladder, and of course the liver kind of is giant compared to it. And here we have the cystic duct, which carries bile to join with the common hepatic duct, and they join together to form the common bile duct, and empties into the main pancreatic duct into the duodenum. Now, one common ailment of the gallbladder is the precipitation of calcium salts and cholesterol to form what are called Gall Stones. Okay, and these may block the ducts and may require removal of the gallbladder. It's known as a cholecystectomy. So here's a picture of the gallbladder with the gall stones that's going to be blocking the movement of the bile. Okay. So in the next slide is just a summary of everything we discussed so far of the accessory structures. Some of this we talked about in the last lecture. Okay, and so the next part of the digestive system, we'll talk about the actual digestion and absorption processes that occur, and then we'll go to the large intestine.